Hello friends, today we are going to discuss one of the important topic about transposons with particular emphasis on lines. That means long interspersed nuclear elements and signs, short interspersed nuclear elements. You know, transposons, transposons, lines and signs. Before going to the topic, we have to understand the basic theme connected to transposons. Recombination. What is recombination? It is nothing but the exchange of genetic information. And it is of two kinds, homologous recombination and non-homologous recombination. Homologous, the word it implied, it is exchange between identical or almost identical chromosomes. Say for example, parental chromosomes, say a 22nd pair of human chromosomes, one from father and one from mother. An exchange between these two chromosomes is known as homologous recombination. There are specific homologous sequences associated with this homologous recombination. But there is another type, non-homologous recombination, which is totally illegitimate recombination. In the sense, it doesn't involve particular specific homologous sequence. Say for instance, transposons and random integration of outside genes into the mammalian genome is also one more example for non-homologous recombinations. That means transposons, otherwise called transposable elements, and the phenomenon is called transposition, comes under the head of non-homologous recombination. Okay, just for understanding, say recombination, it is of two, homologous, then non-homologous. In homologous recombination, say for instance, you consider two parental chromosomes for easy understanding. Say this is from mother and this is from father. That means these two are parental chromosomes. Parental chromosomal pairs. Then here recombination results to, say, then one more type, that means here this has been exchanged between these two, that means one from mother and one from father. A particular region, a particular segment has been exchanged between these two, resulting in homologous recombination. It simply means that, it is implied that our chromosomes are not intact. They are subjected to recombinations like this through concern or respective homologous sequences. But it is all with respect to, because 22nd pair I told example, homologous chromosomes. That means recombination between two homologous chromosomes simply called homologous recombination. But non-homologous is a different kind. Say the best example are transposons. Transposons and outside DNA incorporation into mammalian cells randomly, just randomly. This is about the basic theme of the concept. Then let me focus on the main original topic, transposons. They are nothing but movement of specific pieces of DNA. That means specific pieces of DNA are under mobile phase. That means mobile DNA fragments, they are known as transposons, otherwise called transposable elements, and the phenomenon is called transposition. That means not all the DNA, that means not all the DNA fragments or different regions of DNA in a genome may have this ability. There are specific portions which have this ability. Okay. Depending on the kind, there are two kinds of transposition. The retrotransposition. Number two, DNA transposition. In DNA transposition, again there are two kinds. Conservative transposition, that means conservative DNA transposition, and replicative DNA transposition. The name it implied. Retro means here there is a requirement of an intermediate other than DNA. Here intermediate is RNA. RNA intermediate through which the transposition is driven or has been or initiated. But here in DNA transposition it's not like that. It's direct transposition from DNA to DNA. That's it. There is no other, that means like RNA as an intermediate. Here DNA itself is an intermediate, whereas in case of retrotransposition there is a requirement of RNA for movement. It can move to any point, that means almost any region of the target chromosomes, almost, like for example within the chromosomes or any chromosome, what we call non-homologous loci. This non-homologous loci present in any genome. That means here depending on the kind of transposition, I told you already there are two kinds, and sometimes there is a requirement of duplication, that means another copy of transposons, sometimes it not. Okay, so let me discuss one by one. Say that only this movement of specific piece of DNA is transposition, so called mobile DNA fragments. There are two types, retrotransposition. Number two, DNA transposition. For illustration, say retrotransposition. First, we'll discuss about retrotransposition. Okay. Say this is a specific DNA region. That is a specific region of DNA where a transposable element is present. Okay. This is transposable element. Transposable element. Right? Here, retro means there is a requirement of RNA. How the RNA is, we know that what we have discussed at length in gene expression or transcription. Say, through transcription, it creates an RNA. Transcription. Then, reverse transcriptase. Reverse transcription creates DNA. And this is called another copy of one particular retrotransposon. Here the kind of transposon is called retrotransposon. Okay, and a copy of the same has been generated out of retrotranscription process. That means reverse transcription process. Say mRNA, it's a usual, we are not going to debate mRNA through transcription, again reverse transcriptase, then to get clear DNA. This is how the HIV virus integrates into the host genome. This is very important, and this DNA has to integrate. That means has to occupy a particular region. Maybe the point is where, either within the chromosome, that means within the same chromosome from which it has been generated, or another chromosome. I told you already, within the same chromosome or any non-homologous loci. Consider within the same chromosome. Say here, it is one copy. Actually, it is a parental copy or original copy. Then we have another copy of the same. These are two retrotransposons. Okay, retrotransposons. This is a parental copy. This is a new DNA. Considered in the same chromosome. Sometimes it may move to other chromosome anywhere in the genome, almost any location in the chromosome. I told you already, any location in the chromosome is such an ability, such an extent of ability the transposons possess. Okay, so it's about retrotransposition, how the issue ends. Then coming to the next kind, DNA transposition. It is of again two kinds. Conservative transposition. One more kind, replicative. The word it implied, replicative transposition, means here we are going to generate one more copy of a particular transposable element so that the same can move to appropriate location. Whereas conservative DNA transposition, the name it implied, there is no copy of, that means the additional copy of concerned transposable elements. That means the entire DNA, that means the concern or specific DNA fragment, what we call transposon, shall be removed. What we call excision and reintegration, the two phases, excision by enzymes and reintegration into the appropriate targeted location or targeted loci. That means here the same DNA sequence has been conserved. That is why it is called conserved transposition. But here, that means in duplicate there is one more copy. Two copies of the same DNA within the genome. This is very important. Just like in the retrotransposition, there are two copies. Here in DNA, that means replicative transposition. In replicative DNA transposition, two copies of specific DNA sequence has been present within the genome anywhere. Okay, let's discuss how it ends. About DNA transposition. It means that directed transposition of DNA to DNA. That's it. DNA transposition. First, say this is one, say conserved, and this is replicative. Conserved and replicative. So this is a particular location of a transposon. Is transposable element. T stands for transposable elements. Then through process called excision and reintegration will be transferred to targeted location. So this will be a target. So for clarity, I am drawing like this. Okay. That means this entire part in a particular location, then in a particular loci, has been excision. That means removed, completely removed. It's like completely removing one particular DNA fragment and reintegrating into some other location. That means that means the same DNA sequence has been conserved without any additional copy. But replicating DNA transposition is not like that. Say this is one region, one transposable element, then there is a duplicate, then there is a replication. That means we are creating one more copy of the same, that means replication. And say for understanding, we will think like this, within the same chromosome. Okay, this is one and this is another one. This is a copy or new DNA, and this is a parent transposon. That means there is an exact copy of the same, then it has been integrated into the appropriate loci. I told you, same or anywhere. That means two copies we have. Two copies. That is why replicative, this is only one copy. This is why called conserved. The same sequence has been conserved. Okay. This is about the kinds of, the enzymes involving with these will be synthesized by the genes present within the transposable elements. It's very important. And how, that means what is the significance of transposons? I told you already, lines and signs. With particular reference to lines and signs. You know, lines and signs. Long interspersed nuclear elements. Short interspersed nuclear elements. These are the most important transposons. What kind of transposons are there? These two are retrotransposons. Retrotransposons. The common point between the two is retrotransposons. You know, I told you already, there are different kind of transposons. Here, are these different kinds are same in different organisms? No. For example, in eukaryote, retrotransposition is more common than the DNA transposition. In examination, they may ask you like this: which transposition is more common? Say, in case of eukaryotes, retrotransposition. In last class, we have discovered about gene transfer in recombinant technology. They may ask you which of the following technique is involving in or considered as a direct gene transfer, the direct DNA transfer, say particle bombardment we have discussed. Or either of the four, or then one of the four options will be particle bombardment. This is how they frame the question. Maybe it may be a bit complicated. Just give a simple example. Okay. Here the both, that means lines, long interspersed nuclear elements and short interspersed nuclear elements are both are retrotransposons. That means the first the RNA will be generated to transcription, then reverse transcription and DNA. That means the same copy and the same will be integrated. That means reintegration either in the same chromosome or a non-homologous loci in the genome. Very important point. That means the common point is retrotransposition. The name it implied lines and long and short. Say the size of the base pairs. Say for example, in case of long interspersed nuclear element, it will be around 7,000 base pairs. 7,000 base pairs. With respect to short interspersed, it will be around 100 to 700 base pairs. And most importantly, why we have to discuss about transposons? Not just examination point of view, it's also about evolutionary point of view also. The present human genome has been resulted, that means the majority of the human genome has been resulted because of the accumulation of transposons. Human genome. That means the simple point is our genome, the majority or most of the human genome is nothing but repetitive DNAs. That's it. Repetitive DNA sequences. How? That means here long interspersed nuclear elements and short they are all. The common point is repeated DNA sequences. Repeated DNA sequences. Accumulation of the same resulting to our human genome. That is why, according to Human Genome Project, most of the human genome is a junk DNA, repeated DNA sequences. The precious part, what we call cream of the genome, what we call exact gene bearing or the encoding portions are maybe around 2%. 98%, even not even 2, sometimes it is even less. That means majority of the genome is junk. Junk because of these transposons. Transposons keep on accumulating themselves, creating a large magnum opus. And out of this, these two are important, lines and signs. Not only just in creation of our human genome, but also in diseases point of view also. I told you already, transposons may also contain genes within the DNA. That means in the transposon sequence, gene sequence are also present, and the mutations in the gene sequence of transposons results to various diseases like breast cancer, colon cancer, hemophilia A, leukemia. There are several disorders associated with the mutations in the transposable genes. In that point of, that means in the epidemiological point of view also we have to discuss about this point. Okay, I told you lines means they will be around 7,000 base pairs, and signs reach to 100 to 700 base pairs. They constitute major junk part of DNA. For example, how say long lines constitute around 21.1%. Just imagine lines are considered around 21.1%. And what are lines? I think, but they are non-LTRs. LTR means long terminal repeats. There are different kinds, like L1. For understanding, we'll draw like this: L1, RTE, R2, L, jockey. There are different types, we can say families of lines. Here L1 is very important. Just like the same here, ALU sequence. ALU sequence in case of short interspersed in nuclear elements. Before that, what is it? What are LTRs? Long terminal repeats. What are they? They are nothing but identical copies of DNA which have been repeating for hundreds or thousands of times. I told you already, majority of the DNA is a junk DNA. Junk DNA because of repeat sequence, and repeat sequence because of LTRs, because of lines, because of signs. It's all different ways, different kinds. That's it. That means repeated DNA sequences, say identical, repeated for hundreds to thousand times. And out of this is non-LTR category, which have 7,000 base pairs around, okay, and the different kinds. Say L1 generally associated with hemophilia A. Hemophilia A, genetic disease. I told you there are different genetic diseases. Apart from there are several types of lines associated with various genetic elements. Okay. In case of ALU sequence, say it has been associated with breast cancer, colon cancer, leukemia, and others. How they relate? I told you already. They contain genes, the mutations in the retrotransposon, particularly the signs or lines of particular kind. L1, ALU, because of signs, leads to genetic disorder. That is why most of the transposons, whenever we come across transposons, we have to focus on whether there is any genetic disorder, because major junk is coming from out of them. This is very important. And these are very important. Long, I told already, see LTRs. Okay. And one more topic, when we come across these issues like microRNA. So this is about lines and signs. What are microRNAs? The last theme of today's topic. In next class, on the next coming classes, where we will discuss about gene silencing, gene knockdown technology and others. But for now, on the CRISPR technology also we are going to discuss. MicroRNAs. This is very important. They are small, non-coding RNAs, around 22 nucleotides. 22 nucleotides. Their major role is in RNA silencing or interference and post-transcriptional activities. That means post-transcriptional regulation of gene expression. Okay, here RNA silencing is most important. This is one kind of gene silencing. That means regulation of gene expression in a negative way. Here, gene silencing is different one. We are regulating certain genes expression. Here, RNA silencing is one kind, and what is RNA silencing? You know, regulation in a negative way. That means what we call mRNA degradation. You know, next class we're going to discuss extensively about gene silencing, but for now I would like to focus on this particular topic because of examination point and also concept oriented point of view. MicroRNA, they play an important role. They're just small RNA molecules of around 22 nucleotides, that's it. But they have an essential role in what we call RNA silencing or RNA interference. You know what exactly RNA silencing? That is one kind of gene silencing. What is gene silencing? It is nothing but the regulation of expression of certain genes. Technically, gene silencing is similar, analogous to gene knockdown technology. It's an experimental technology, okay? And we will discuss about CRISPR and others, okay? But and this one, that means RNA silencing or RNA interference is one category of gene silencing, and they play an important role in negative regulation of gene expression. What is negative? Simply mRNA degradation. It is a way of gene regulation of microRNAs. Okay, this is for today, and for next class we'll go to discuss about